piece off right here to get down into where the blood is. Fingertips and toes are saturated with blood. And I had the DNA tested, and it is human. And there's fingerprints. You see that? That's the fingerprint I broke off the side. These are the sweat pores. See the sweat pores? This, and it broke right off, like it came right off. It's like the layer underneath it came off just like Velcro, just like that. Like this is like a rubbery piece, and you can't get any blood out of that. That's why I did it out there. I know these things inside. Now, I got them all over my property. And I'll show you another one. I got a hand out there that's almost 4 feet wide. This is 3 feet long. They're just a fingertip, just the tip of the finger. The other one, the hand itself, is 4 feet wide. And it's, anyway, and that was DNA tested too. I'll show you the DNA test. But this is, my finger is about the width of one of the ridges in your fingerprint. All right, this is gigantic. I've calculated I was about 150 feet tall, this guy would have been, if everything anatomically was exact, and it appears to have been. All right, here's another one, goes back to 2015. I was the first one in the world, and so was the Helix Biolabs, to test for human DNA in ancient rocks. I extracted the blood samples, and I sent them to Helix. They did not do the extraction. They did the analysis of what I sent them. I sent them 3 mud fossil samples, and they extracted DNA from the samples I sent them. They did not extract them from the actual complete mud fossils. And they did PCR DNA sequencing and analysis. And the objective was an analysis to perform extraction of any genomic or mitochondrial DNA from these mud fossils. Upon successful DNA extraction from the sample, subsequent analysis will include the amplification of extracted DNA using PCR technology, targeting specific DNA marker sequences to obtain DNA sequence from the amplified PCR process. What does that mean? They are going to look at DNA if they can find it, and they did, and they found it was excellent quality and it was dense, because I know where to take it from. I took it right out of the blood. I literally sent them almost blood that was dry. Now, so what did they do? They checked it to see what the sequences were in this DNA. And if it had these exact targets, you see targets? Your DNA's got bazillions of particles. Now, there's sequences that determine what, if you're a Homo sapien or you're a zebra or whatever you are. And if they can find that set of sequences, because there's still about, let's say, I'm just throwing a number. Let's say there's 500 particles in a row that are exactly the right particles. That's not going to happen accidentally. That means you are the thing that 500 particles represents, which is Homo sapien in this particular case. So what they did was they took out those particles, and then they had them all figured out, and I don't know, they did a lot of testing on them and so forth, and then they sent them off, and they used all very, very serious stuff. Because this is the first in the world. I worked with a guy at the lab there, and I said, Tom, his name was Tom. I said, Tom, this is first ever. I mean, can you do it? He said, well, we don't know. He said, nobody's ever done it before, but we can try this. So they did this paper following the procedure. He said, I found procedures that might work with this. And I said, well, do what you can do. And I paid him. And it wasn't cheap, but it wasn't that expensive either. Well, it was expensive, but it wasn't, thousands and thousands of dollars. It was enough I could pay for, let me put it that way. All reactions involve 1 primer per reaction. In some instances, better quality amplification of the target mitochondrial DNA was achieved using DNA from the second elution. So in other words, they did it on different ways to get different concentrations and densities. But whatever they did, when they got it done, it ended up being successfully tested for mitochondrial DNA. That means the mother side. So, and we all have the same mitochondrial DNA, which is a mother side. And it comes down to here, where they say PCR product is submitted to Eaton Biosciences in New Jersey. Now, this is the one I had, Helix Biolabs. They did the extraction of the DNA sequencing, and they sequenced it. All right, or in New Jersey for DNA sequencing, the primer pairs that were used to generate the PCR products were also in DNA sequencing. Excellent quality DNA sequence was obtained for that gigantic fingertip, the long one, it's almost 36 inches long. And the long one, here's the long, hold on. Because these were found basically in the same hole. That lung is our size, and that lung is again perfect, very, very dense blood came out of that tip, because that's where the blood collects at the tip. And so anyway, this was all sent off, and it came back Homo sapien mitochondrial, cytochrome B gene, and D loop region. Both of them mitochondrial, all in Homo sapien. And here's all the CTAGs, and you know, this is all the sequences in the DNA that shows and proves what it was. You don't get all that stuff in an order like that and have it just be accidental. All right? And it's a pretty serious test. And again, this was the first in the world that was ever done. And I talked to Tom about this because he was getting attacked by people, you know. Anyway. All right, I don't want to go down that road. But anyway, here's what they ended up having. They submitted these DNA sequencing to a blast search using the National Center for Biotechnology Information, NCBI database, which apparently has every known sequence of DNA. So it says whereby each DNA sequence was matched to all DNA sequences contained in the NCBI DNA sequence database. So I don't know how many there is, but all. Now the results of that blast search, so they just said, see if you can find any sequences that match these using the DNA sequences they generated from these things that I sent them. Indicates the Homo sapien might go. Now, if I'm wrong, they have to prove I'm wrong. Anatomists say yes, they're identical to a human lung. It even has the depression of the heart because it's a left human lung. And the heart would sit right there as this guy died, flat in the flood. And that's why it's so flat like that on the back. Now, this is what also upsets them terribly because it totally wipes out evolution. I'm showing you giants that they had talked about in the Bible, in the ancient texts. Every one of them talks about giants. Every culture on earth talks about giants. 100%. 100%. And they also talk about dragons. 100%. And I have found them very, very, very large dragons. This is what upsets academia. And until they will address that, they have gone into a range where they are not doing their job.
Bro,